This is prophase, the first division of mitosis. Now, here we are inside the cell. Here you can find the two asters. This is the aster. And in between, you can find the spindle fibers. And here, if you look inside the nucleus, so this is the representing the nuclear membrane. So this is all this round is the nucleus, and the membrane of the nucleus, which is called nuclear membrane, start to disappear, start to dissolve. Okay, and inside the nucleus you can find the 46 chromosomes, because we are talking about a human cell now. Here, of course, if you look, for example, at one chromosome, okay, so this is a chromosome connected here by a centromere, and of course this are the sister chromatids, which are exactly copy of each others. The second phase, metaphase. What happens during metaphase? As you can see here, the asters just move to the opposite poles here and there, and here you can see that all the 46 chromosomes just line up in the middle of the cell, or align in the middle of the cell to form what? The equatorial plate. Exactly. So you can see that all the 46 chromosomes just line up in the middle of the cell, noting that in a way that the chromatids just are opposite to each other, and you can see the centromere in the middle, and all the spindle fibers that extended from the asters, they attach the chromosomes at the centromere from the two asters, so that they are attaching all the chromosomes, all the 46 chromosomes from their centromeres. And as we said, the lining up of these 46 chromosomes just form the equatorial plate. This is Anaphase now, the third phase of mitosis. Two things happen during Anaphase, which are the separation and migration of the sister chromatids. As you can see here, all the sister chromatids of the 46 chromosomes separated from each other. Here there are two sister chromatids, separate from each other from the centromere, right? So all the 46 chromosomes, they now becoming two sister chromatids apart from each others. And if you look at it from a different angle, you can see here the space, which means that they are totally separated from each others, and you can notice the centromeres as well. So what will happen next is that each aster will pull these spindle fibers towards it, right? So that all these chromatids will go to this side, and all these chromatids will go to the other side, so that we are separating totally the sister chromatids, and they migrate, or they move away from each other. As we have seen in the picture, they were lining up here in the middle of the cell. Then the sister chromatids separated from each other. Then each chromatid, they migrate away from each other. This one goes to this one, to this place here, and the second chromatid moves to this pole here. So as you can see, there is a sister chromatid here and a sister chromatid here. They go away from each other, as you can see, and they are attached from the centromere. They move away to the opposite poles. And also, if you notice here, when the chromosomes separate from each other, now we obtain a chromosome of one sister chromatid. So at the beginning, they were a chromosome of two chromatids, okay? And then when they separated from each other, they become a chromosome of one chromatid. So this chromosome of one chromatid go to one pole, and this chromosome with one sister with one chromatid with one capital N go to this pole. So this is anaphase. Telophase, the fourth phase and the last one of mitosis, which is the division of the two cells and obtaining the two daughter cells. What happens in each cell is that the 46 chromosome of one chromatid of one capital M, they just now with each other, and they are surrounded by the nuclear membrane. We can see here the appearance of the nuclear membrane. We can see that the asters, they transform back to centrosomes, and of course you can see that there are no spindle fibers anymore, right, in here. And also the same thing happens in the second cell. And what happens in between, now this is the constriction of the cytoplasm, or the cleavage of the cytoplasm from the two sides. You can see, so this one was one big cell, and now it's splitting into two, so that we are obtaining two identical daughter cells. Of course here, why they are called identical cells? Because the number of chromosomes here are exactly the same number of chromosomes here, and also they are exactly the same copy of this chromosome. For example, if we see here, this is a chromosome with one chromatid, we must find the same one here as well. So this is the sister chromatid. It's exactly the same. Let's, for example, take another one. This one, for example, this is a chromosome with one chromatid, and of course we will see the same one here as well. So that the 46 chromosomes with one chromatid here, we can find exactly the same in the other cell. And we say that these two cells are exactly identical. Of course, what happens next? This one will just be splitting completely, obtaining two identical daughter cells. And this is the end of telophase and the end of mitosis.